welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we are looking at knowing how a triglyceride is synthesized by the formation of ester bonds during condensation reactions between glycerol and 3 fatty acids. And we are also going to be looking at the difference between saturated and unsaturated lipids. So what are lipids? Lipids are macromolecules that contain carbon, hydrogen, and oxygen atoms. And what you would have noticed is that that's exactly the same elements involved in glucose. So what is the difference between lipids and carbohydrates when they literally have the same elements? It's that lipids contain a lower proportion of oxygen. Lipids are non-polar and hydrophobic, which means they are insoluble in water. And an example of lipid is triglyceride. It forms the main component in fats and oils. So now, looking closer at triglycerides, they are non-polar and hydrophobic molecules. Triglycerides basically being lipids, it is insoluble in water as well. The monomers within triglyceride are glycerol and fatty acids. And this is very, very important to know. Once again, you just like how you know how to draw out the molecule of glucose, you need to know how to draw out glycerol and fatty acids because it might come up, and it's just good to know. So glycerol goes through a condensation reaction with 3 fatty acids, as you can see in this diagram, and like all condensation reactions, you will get 3 molecules of water at the end of this reaction alongside a triglyceride. So the shorthand chemical formula for a fatty acid is RCOOH, where the R can be any long chain of carbon. So now, function of lipids. Lipids functionality revolves around energy yield, energy storage, insulation, and hormonal communication. Triglycerides being fats and oils, we can look at the role of fats and oils in the body too. So once again, what is the role of triglycerides? Energy storage, just like lipids. Insulation, once again, just like lipids. Buoyancy and protection. So now, looking at formation of an ester bond. So just a quick throwback to a previous video, the bond between 2 glucose molecules in a carbohydrate chain would be glycosidic bonds. So similarly, the bond between glycerol and the 3 fatty acids involved is an ester bond. So that is what we're going to be looking at right now. So triglycerides are formed by a process called esterification, which is a type of a condensation reaction. And like I had said earlier, 3 water molecules are released at the end of this. The bond between glycerol and fatty acids is known as an ester bond. So now, types of fatty acids in lipids. So fatty acids can differ in 2 ways. So one way it can differ is the length of the hydrocarbon chain, and another way it can differ is the fatty acid chain, being saturated or unsaturated. So saturated fatty acids contain no carbon-carbon double bonds, giving rise to unbranched linear chains. So they have no double bonds between them. And unsaturated fatty acids can have double bonds in the hydrocarbon chain, but once again can differ in either mono or poly. So mono is when there's one double bond in the hydrocarbon chain, and poly is when there's multiple double bonds in the hydrocarbon chain. And just to point this out, the double bonds we are talking about for saturated and unsaturated fatty acids is focusing on the double bonds in the hydrocarbon chain alone, not anywhere else, like not in the oxygen or something like that. So if you get one of those questions where they ask you which of the following is an unsaturated fatty acid, make sure you're looking for double bonds in the hydrocarbon chain, not anywhere else. if it's anywhere else, it could still be a saturated fatty acid. So now, cis fatty acids and trans fatty acids. If the hydrogen atoms on the same side of the double bond, the fatty acids would be cis fatty acids. They can be metabolized by enzymes. So this is something you might have come across in A-level chemistry if you've done the topic so far, but the hydrogen atoms of the double bond, so attached to the carbons in the double bond, if they're on the same side, it means they are cis fatty acids. And if they're on opposite sides, then they would be trans fatty acids. So now, if the hydrogen atoms are on opposite sides of the double bond, these would be trans fatty acids. They cannot form enzyme substrate complexes, hence not metabolized. They are harmful for your body and increases the risk of developing coronary heart disease. So trans fatty acids are the ones you want to avoid. So that is all from Professor Snap today, and hope you have found this video useful. If you have any questions or if you need any clarification on what we've discussed, please let me know in the comments below, and share this to someone who might find this useful, and thank you for watching.